All right, folks, let's go over what you need to study for our test on the cell cycle. We have one set of foldable notes for this unit. It should be in your VIP section, and this is the cell cycle foldable. On the front, you've got the steps of mitosis, and on the back, you have a flap for gamete reproduction, aka meiosis. Some people say meiosis, and a flap for binary fission, which is the way that simpler organisms like prokaryotes reproduce asexually. In our labs and project section, we have this sheet, which is a modification from the Explore Learning gizmo. It is on the cell cycle. On the back, you drew the pictures of the phases and you filled in some blanks. And as always, you've got your portfolio and your glossary for this unit. Please note at the bottom, the glossary says that you need to know all of the stages of the cell cycle, including the steps of mitosis, in the order that they occur. You also need to be able to identify them based on a picture, especially if that picture is not presented necessarily in order. As always, if you check my website on Phoenix, you can find full copies of the foldable notes, the class presentation, as well as blanks if you need to print a new one. You can also find several review game links. I've made a lot of Quizlets for this unit, including a fill-in-the-blank sentences one and a lot of picture matching using real microscope images that I think will be very helpful. You can also find links to this and other review videos by visiting my Phoenix website. So let's start by talking about what on earth the cell cycle is. The cell cycle is basically the life cycle of a cell. A cell spends most of its life in interphase. A little bit of its life is spent copying its nucleus, and an even smaller amount of its life is spent splitting up that nucleus. The phase of mitosis, which is in eukaryotic body cells, can be further split down into 4 stages: prophase, metaphase, anaphase, and telophase. All of this is, of course, another fabulous circle of life. So like we said, interphase is when a cell is just living its life. In this phase, a cell might be doing endo and exocytosis, it might be doing osmosis, other forms of passive transport, it's making proteins, and it's basically doing whatever it's supposed to do. If it's a muscle cell, it's helping move a body. If it's a digestive cell, it's helping with digestion. If it's a blood cell, it's carrying oxygen. Basically, whatever life that cell is meant to live, it's happening in interphase. The 2 phases of mitosis and cytokinesis are so short that most of the time when you see a picture of a cell or think of a cell in general, it's happening in interphase. Let's look in a little greater detail at each of these phases, and we'll start with interphase. So like we said, a cell in interphase is just living and loving its life. But it might get a signal that, hey, you know what, cell, you're getting kind of old, it's time that you're replaced. Or, you know what, cell, we need to heal, or we need to grow in this body. So the cell will get a signal, perhaps from an outside source, or perhaps from hormones or other chemical signals, to start doubling. So it's going to double everything in its nucleus. Today we're going to focus mainly on what's happening in the nucleus, but everything would need to be copied. The mitochondria, the ribosomes, the endoplasmic reticulum, the Golgi complex, all of that would need to be copied. But here we've got twice as much of the nuclear stuff, the DNA, inside that nucleus right at the end of interphase. Now would be a great time to pause and talk about the differences between DNA, chromosomes, chromatin, and chromatids, because all of those terms are probably super confusing. So here we have a chromosome, we have some sister chromatids, we have DNA, and we have chromatin. Let's talk about how all of these things are related. Hopefully it will clear up some of the confusion. So this X shape is a chromosome. When you see an X shape, you can think, okay, I see an X. That's like a multiplication symbol. That means that the cell is getting ready to multiply. Biology, you'll hear the terrible joke, is the only science in which multiplication is actually division, because the only way that a cell can make more copies of itself is to divide. So a chromosome, when it splits during anaphase, we'll talk about that phase in a moment. When it splits during the phase of mitosis called anaphase, it forms sister chromatids. These are identical copies of each other. The chromosome is simply the chromatids joined by a structure called a centromere. The sister chromatids are made up of coils of this structure called chromatin. Chromatin is a bunch of coils of proteins and DNA. Woo! You'll be making a keychain of DNA in our next unit. Little teaser trailer there. So to recap, if I have a lot of DNA and I condense it down with some proteins, I make chromatin. If I condense that chromatin down, I make chromatids, which are equal copies of one set of genetic information. When these are joined together by a centromere, I have a chromosome. All of this doubling stuff is happening right at the end of interphase. After interphase, the cell is going to go into prophase. Prophase is go phase. During prophase, the cell has chromosomes looking like that X shape. Each chromosome is 2 copies of identical genetic material. Why do we need to have copies during prophase? Well, it'd be like if I gave you a very majorly important homework assignment. If you only do one homework in your life ever, it should be this one. Do not lose this paper. And your table mates did not get any copies of this worksheet. Well, you might think, oh, no problem. Here, I'll just tear this in half. Now you have a copy of the important worksheet and I have a copy of the important worksheet. But as we know, that's not going to fly. If you only have half a set of this important worksheet, if you only have half of the info that you need, and yes, of course, there's writing on the back because we're not going to waste trees, you're not going to be able to be successful. Likewise, in order for a cell to be successful and successfully divide, it needs to copy everything so that when it splits, 1/2 can go in a fair split to each new cell. So mitosis officially begins in prophase. Prophase is go phase. It's the first phase of mitosis. Here's our life cycle of the cell. Here's that split of mitosis. Right after prophase, which is go phase, you enter metaphase. You can remember metaphase like middle phase because in metaphase, all of the chromosomes are lining up in the middle of the cell. It's like when you're moving and you really need to get organized. You have everything and you're going to put it in organized boxes. And as you know, if you've ever had to move, moving is a very stressful process. So you're going to want to put everything that's important to you in an organized container so that that move is easier. That's what's happening during these phases of mitosis. After metaphase, that middle phase, comes a phase called anaphase. Anaphase is the apart and away phase. It's our third phase of mitosis. In this phase, the sister chromatids, which were joined by a centromere, are going to split apart. Recall that when they were in metaphase, they were all lined up in the middle, like a tug of war. These spindle fibers attached to the centrioles have pulled so hard that the sister chromatids split and one goes to either side of our cell. Just like in a globe or on a map, we have our equator, where all of those chromosomes line up, and we have our poles. The spindle fibers, which are attached to the centrioles, are pulling the chromatids to the poles during anaphase, the apart and away phase. After anaphase comes telophase. Telophase, you can remember like T for 2 because suddenly we have 2 nuclei. The sister chromatids, pulled by the spindle fibers attached to the centrioles, have moved from the equator to the poles of the cell. We now see the nuclear membrane again in 2 places because we've copied the nucleus. We are almost ready to split into 2 cells, but we are not yet 2 cells. We have 2 nuclei, but we're not yet 2 cells. At this point, however, because we've copied the nucleus, mitosis is all done. The final phase in a cell's life is cytokinesis. This is when, after telophase, the cytoplasm is going to get split, creating 2 identical daughter cells. These daughter cells are identical to each other and identical to the original parent cell that we met in interphase. Once a cell has gone through all of these phases, interphase, all of the steps of mitosis, and cytokinesis, its life cycle has completed. So all the cells I just showed you, these were animal cells. We know that they were animal cells because they have centrioles. Plant cells do not have centrioles, so they do cytokinesis, that final step. They do this a different way. When a plant cell transitions from telophase to cytokinesis, it kind of builds a temporary cell wall called a cell plate. You can kind of think of that as like the dividers that are in some of the math classrooms. They used to be resource rooms, but now the dividers are pulled up to make 2 new smaller classrooms. Eventually these 2 cells will grow and become bigger, but for now they've got this temporary cell plate that will build up to be a stronger cell wall during cytokinesis for a plant cell. So we also learned this really cool secret handshake of the cell cycle, and I will review it with you right now. So we started with our hands like this in interphase. I said this is kind of like making a capital I for interphase. This is when the cell is just hanging out, doing its cell thing, living its life, making proteins, doing passive and active transport, living the cell life. In prophase, those chromosomes are going to become visible, everything's going to condense down, and we're going to lose sight of the nucleus. Everything has doubled at this point. In fact, it all copies at the end of interphase when the cell gets that signal, hey, it's time to divide. But prophase is go phase. Mitosis has officially begun. Then in metaphase, we have all of the chromosomes lined up in the center of the cell, or the equator. My fingers here kind of look like the X's of those chromosomes lined up in the equator. In anaphase, these separate, they go apart and away to the opposite poles. Telophase, we draw 2 new nuclei. Mitosis is complete because mitosis is copying the nucleus. And cytokinesis, the cytoplasm splits apart to make 2 new daughter cells. So altogether, that's interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. So to recap, a cell's life cycle is mostly interphase, followed by a brief mitosis and an even briefer cytokinesis. Mitosis can be split into 4 main steps: prophase, metaphase, anaphase, and telophase. The result of this whole process is 2 identical cells. But mitosis is not the only way that cells can divide. There are other ways that cells can divide. One of these other ways that cells can divide is called binary fission. Binary fission is a process done by prokaryotes, archaea, and bacteria. These are cells that are more simple and they don't have a nucleus to copy. Their DNA is in a big tangly loop. So in binary fission, you start with 1 cell. The cell doubles its genetic content and begins to split into 2 new cells that have an equal amount of DNA as the cell that started out. This is very different from the mitosis process that we saw in eukaryotic cells. In eukaryotic somatic cells, the cells that have a nucleus and have organelles, so animal cells, fungus cells, protist cells, plant cells, all of these cells, if we're talking about their body cells, will do mitosis. Prokaryotes, such as archaea and bacteria, do binary fission. But wait, there's more. There's one other way that we talked about in this unit that cells can divide. If we are talking about gametes, which are reproduction cells, these cells undergo a process called meiosis. And I'm going to demonstrate meiosis using cats. Meiosis is the process, you'll sometimes hear people say meiosis. It's the process that eukaryotic cells use to ensure that the next generation has a unique genome. So let's say you have 2 cats. You can see that these cats don't look really anything alike. But let's imagine that these cats had a cat baby. Each parent would contribute some of their DNA to create a unique organism. If these 2 cats had a baby, it might look like this. It has some traits from one parent. You can see the gray fur and the kind of round face. It has some traits from the other parent, such as the big green eyes. And then there's some traits that we didn't see in either parent, like these rounded ears. This new organism is incredibly unique, like a snowflake. No other organism has its exact DNA unless it's an identical twin. Some of its genes came from one parent and some of its genes came from another parent to create a unique organism. The main difference between meiosis and mitosis is that mitosis creates 2 identical somatic body cells that are identical to the original cell. Meiosis creates 4 unique gametes, or reproductive cells. That's the extent of what you need to know for this unit. In our next couple units, we'll talk specifically about the structure of DNA that allows these differences to happen, and we'll talk a little bit more about how 2 very different looking organisms can create a unique organism when we study our genetics unit.
If I have a lot of DNA and I condense it down with some proteins, I make chromatin. If I condense that chromatin down, I make chromatids, which are equal copies of one set of genetic information. When these are joined together by a centromere, I have a chromosome. All of this doubling stuff is happening right at the end of interphase. A cell's life cycle is mostly interphase, followed by a brief mitosis and an even briefer cytokinesis. Mitosis can be split into 4 main steps: prophase, metaphase, anaphase, and telophase. Our steps in order are interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Altogether, that's interphase, prophase, metaphase, anaphase, telophase, and cytokinesis.